Continuation, it's about B lymphocytes, B cells. The B lymphocytes, also called the B cells, derived its later designation from its site of maturation in the bursa of Fabricius in birds. The name turned out to be apt, as bone marrow is its major site of maturation in a number of mammalian species, including humans and mice. Mature B cells are definitely distinguished from other lymphocytes and all other cells by their synthesis and display of membrane-bound immunoglobulin molecules which serve as receptors for antigen. Each of the approximately 1.5 into 10 raised to 5 molecules of antibody on the membrane of a single B cell has an identical binding site for antigen. When a naive B cell, one that has not previously encountered antigen, first encounters the antigen that matches its membrane-bound antibody, the binding of the antigen to the antibody causes the cell to divide rapidly. Its progeny differentiate into effector cells called plasma cells and to memory B cells. So memory B cells have a longer lifespan than naive cells and they express the same membrane-bound antibody as their parent B cell. Plasma cells, on the other hand, produce the antibody in a form that can be secreted and have little or no membrane-bound antibody. So plasma cells are end-stage cells and do not divide. Although some long-lived populations can be found in bone marrow, many die within 1 to 2 weeks. They are highly specialized for secretion of antibody, and a single cell is estimated to be capable of secreting from a few hundred to more than 1,000 molecules of antibody per second. Then T lymphocytes or T cells. T lymphocytes derive their letter designation from their site of maturation in the thymus. During its maturation within the thymus, the T cell comes to express on its membrane a unique antigen presenting molecule called the T cell receptor. Unlike membrane-bound antibodies on B cells, which can recognize antigen alone, T cell receptors only recognize antigen that is bound to cell membrane proteins called major histocompatibility complex molecule. MHC molecules that function in this recognition event, which is termed antigen presentation, are genetically diverse, polymorphic glycoproteins found on cell membranes. There are two major types of MHC molecules. Class 1 MHC molecule expressed by nearly all nucleated cells of vertebrate species, and class 2 MHC molecule which are expressed by only a few cell types that are specialized for antigen presentation. So when a T cell recognizes antigen combined with an MHC molecule on a cell, under appropriate circumstances, the T cell proliferates and differentiates into various effector T cells and memory T cells. Then there are two well-defined subpopulations of T cells, which are T helper cells, TH cells, and T cytotoxic T cells. Recently, a third T subpopulation, T regulatory cells, has been characterized. T helper and T cytotoxic cells can be distinguished from one another by the presence of either CD4 or CD8 membrane glycoproteins on their surfaces. T cell displaying CD4 generally function as TH cells, whereas those displaying CD8 generally function as TC cells. Thus, the ratio of TH to TC cells in a sample can be approximated by assigning a number of CD4 plus and CD8 plus T cells. This ratio is approximately 2:1 in normal human peripheral blood, but it may be significantly altered by immunodeficiency disease, autoimmune disease, and other disorders. Following activation by interaction with appropriate antigen MHC complexes, TH cells differentiate into effector cells that enable or help the activation of B cells, TC cells, macrophages, and various other cells that participate in the immune response. Alternatively, some TH cells differentiate into memory cells instead of effector cells. So, recognition of antigen MHC complexes by TC cell triggers its proliferation and differentiation into an effector cell called a cytotoxic T lymphocyte or CTL, or into a memory cell. The CTL has a vital function in monitoring the cells of the body and eliminating any that display foreign antigen complex with class 1 MHC, such as virus infected cells, tumor cells, and cells of a foreign tissue graft. T regulatory cells are identified by the presence of both CD4 and CD25 on their membranes. However, unlike a CD4 bearing T helper cells, T reg cells suppress immune response. They are negative regulators of the immune system. Like TH and TC cells, members of the T reg subpopulations of T cells may be progenitors of memory cells. Then B and T populations comprise subpopulations of clones. All of the antigen receptors on a given B or T cell have identical structures and therefore have identical specificities for antigen. If a given lymphocyte divides to form two daughter cells, both daughters bear antigen receptors with antigen specificities identical to each other and to the parental cell from which they arose, and so will any descendants they produce. The resulting population of lymphocytes, all arising from the same founding lymphocyte, is a clone. At any given moment, a human or a mouse will contain tens of thousands, perhaps at 100,000 distinct T and B cell clones, each clone distinguished by its own distinctive and identical cohort of antigen receptors. Contact with the antigen induces these cells to proliferate and differentiate. The products of this process include both effector cells and memory cells. The effector cells carry out specific functions while the memory cells persist in the host and on re-challenge with the same antigen mediator respond, that is only both quicker and greater magnitude. The first encounter is termed a primary response and the re-encounter a secondary response. Next is about natural killer cells. Then we will be learning about granulocytes, neutrophil, basophil, some stuff like that. Then dendritic cells, organs of immune system like thymus, bone marrow, lymphoid system, secondary lymphoid organs in quick, spleen in particular, then mucosa associated tissues like that. So we'll be quickly skimming through all these points. The body contains a small population of large granular lymphocyte cells called the natural killer cells that display cytotoxic activity against a wide range of tumor cells and against cells infected with some but not all viruses. An extraordinary feature of these cells, which constitute 5% to 10% of lymphocytes in human peripheral blood, is their ability to recognize tumor or virus-infected cells despite lacking antigen-specific receptors. Natural killer cells are part of the innate immune system, and most do not have T cell receptors or immunoglobulin incorporated in their plasma membrane. In other words, they do not express the membrane molecules and receptors that distinguish T and B cell lineages. NK cells recognize potential target cells in two different ways. In some cases, an NK cell employs NK cell receptors to distinguish abnormalities such as reduction in the display of class 1 MHC molecules or the unusual profile of surface antigens displayed by some tumor cells and cells infected by some viruses. In addition, some tumor cells and the cells infected by certain viruses display antigens against which the immune system has raised an antibody response, so that anti-tumor or antiviral antibodies are bound to their surface. Because NK cells express a membrane receptor CD16 for a specific region of the antibody molecule, they can attach to these antibodies and subsequently destroy the target cell. This is an example of a process known as antibody-dependent cell-mediated cytotoxicity. What is happening? Because NK cells express a membrane receptor for a specific region of the antibody molecule, they can attach to these antibodies and subsequently destroy the target cells. NK cell cytotoxicity, the focus of much current experimental study, is described. Recognition has recently been growing of another cell type, the NKT cell, which has some of the characteristic of both T cell and NK cell. Like T cell, NKT cells have T cell receptors. Unlike most T cells, the TCRs of NKT cells interact with MHC-like molecules called CD1 rather than the class 1 or class 2 MHC molecule. Unlike NK cells, they have variable levels of CD16 and other receptors typical of NK cells, and they can kill target cells. A population of activated NK cells can rapidly secrete large amounts of the cytokines needed to support antibody protection by B cells, as well as inflammation and the development and expansion of cytotoxic T cells. Inflammation and the development and expansion of cytotoxic T cells, determining the exact roles played by NK cells in immunity, is a great interest. Then mononuclear phagocytes. The mononuclear phagocytic system consists of monocytes circulating in the blood and macrophages in the tissues. During hematopoiesis in the bone marrow, granulocyte monocyte progenitor cells differentiate into promonocytes, which leave the bone marrow and enter the blood, where they further differentiate into mature monocytes. So monocytes circulate in the bloodstream for about 8 hours, during which time they enlarge, They then migrate into the tissues and differentiate into specific tissue macrophages. So differentiation of a monocyte into a tissue macrophage involves a number of changes. The cell enlarges 5 to 10 fold. Its intracellular organelles increase in both number and complexity, and it acquires increased phagocytic ability, produces higher levels of hydrolytic enzymes, and begins to secrete a variety of soluble factors. So macrophages are dispersed throughout the body, like take up residence in particular tissues, becoming fixed macrophages, whereas others remain mobile and are called free or wandering macrophages. Free macrophages travel by amoeboid movement through the tissue. Macrophage-like cells serve different functions in different tissues and are named according to their tissue location, like intestinal macrophages in the gut, alveolar macrophages in the lung, histiocytes in the connective tissue, Kupffer cells in the liver, mesangial cells in the kidney, macroglial cells in the brain, and the osteoclasts in bone. So macrophages are activated by a variety of stimuli in the course of an immune response. Phagocytosis of particular antigens or contact with receptors that sense molecules present in microbial pathogens often serve as an initial activating stimulus. However, macrophages activity can be further enhanced by cytokines secreted by activated T cells and by mediators of the inflammatory response. Inactivated macrophages are more effective than resting ones in eliminating potential pathogens for several reasons. They exhibit greater phagocytic activity and increased ability to kill ingested microbes, increased secretion of inflammatory mediators, and an increased ability to activate T cells. In addition, activated macrophages, but not resting ones, secrete various cytotoxic proteins that help them eliminate a broad range of targets, including virus infecting cells, tumor broad range of cells, and intracellular bacteria. The next, phagocytosis is followed by digestion and presentation of antigens. Macrophages are capable of ingesting and digesting exogenous antigens such as whole microorganisms and insoluble particles, and endogenous matter such as injured or dead host cells, cellular debris, and activated clotting factors. Phagocytosis is initiated by the adherence of antigen to the macrophage cell membrane. Complex antigens such as whole bacterial cells or viral particles tend to adhere well and are readily phagocytosed. So, isolated proteins and encapsulated bacteria tend to adhere poorly and are less readily phagocytosed. Adherence induces membrane protrusions called pseudopodia. So, complex antigens such as whole bacterial cells or viral particles tend to adhere well and are readily phagocytosed. Isolated proteins and encapsulated bacteria tend to adhere poorly and are less readily phagocytosed. Adherence induces membrane protrusions called pseudopodia to extend around the attached material. Fusion of this pseudopodia encloses the material within a membrane-bound structure called a phagosome, which then enters the endocytic processing pathway. In this pathway, a phagosome moves towards the cell interior where it fuses with the lysosome to form a phagolysosome. So, lysosomes contain a variety of hydrolytic enzymes that digest the ingested material. The digested contents of the phagolysosome are then eliminated in a process called exocytosis. The macrophage membrane has receptors for certain classes of antibody. If an antigen like bacterium is coated with the appropriate antibody, the complex of antigen and antibody binds to antibody receptors on the macrophage membrane more readily than antigen alone, and phagocytosis is enhanced. In one study, for example, the rate of phagocytosis of an antigen was 4,000-fold higher in the presence of specific antibody to the antigen than in its absence. This antibody functions as an opsonate, a molecule that binds to both antigen and phagocyte, thereby enhancing phagocytosis. The process by which opsonins render particulate antigens more susceptible to phagocytosis is called opsonization. Although most of the antigen ingested by macrophages is degraded and eliminated, experiments with radio-labeled antigens have demonstrated the presence of antigen peptides on the macrophage membrane. Phagocytosed antigen is digested within the endocytic processing pathway into peptides that associate with class 2 MHC molecules. These peptide class 2 MHC complexes then move to the macrophage membrane. This processing and presentation of antigens were critical to Th cell activation, a central event in the development of both humoral and cell-mediated immune response. Then there are some other granulocytic cells.